Good afternoon and welcome to this session about innovation on ECMO, and I'm really happy to see you here. And we're going to start with a perfusionist from Colombia named James Beck. And he's so welcome to come up. Thank you.
Hello. Can everybody hear me? All right, good. Well, welcome back after lunch. If you doze off after that wonderful lunch, I will not be offended, I promise you. I just wanted to thank the organizers of the meeting for inviting me to share some of my thoughts and some of our experiences around innovation today, and thank all of you for your attention this afternoon. The subject that I was asked to speak about, next frontiers and some of that innovation, I have no financial disclosures, but we do currently use several of the Spectrum products. I will be talking a little bit about that. I will also be showing some pictures of things that may or may not be FDA approved and CE marked, so I want to disclose that right off the get-go, things that may or may not actually come to market. You know, I certainly don't have to talk to this group about the past of ECMO, but as you've heard from so many before me, and you know, I know that I probably don't have to say this, but I could not come up and give an ECMO talk without paying homage to Dr. Bartlett and all the work that he's done for all of us. I know in my short 33-year career, started with being guided by Dr. Bartlett and being up here talking about the future of ECMO and still being guided by his tutelage is just an absolutely amazing feat. So thank you for all you do for us. The president of ECMO right now is definitely changing, and you know there's been quite a bit of ECMO programs opening up since the pandemic. And certainly the resurgence of ECMO even before the pandemic has been quite striking. So when we think about what's next or what the future might look like, I think it was Peter Drucker that said, you know, if you want to predict the future, the best way to do that is to create it. So I think it's really incumbent on all of us to create what that future may actually look like. Now, I'm not 100% sure of what it's going to look like or how to predict that, but I am pretty sure that there'll be quite a bit of data acquisition, analysis, and feedback involved in what the future of not only ECMO looks like, but many aspects of the medical community and the medical care that we offer patients, just like it's been used so widely in other industries. Now, as I thought about what the future may look like, the three things that came to mind were certainly new hardware, new disposables, and then new data management, trends, analytics, informatics, and how we can use some of that to help us do a better job taking care of folks. Now, certainly in the 10 minutes that I have, I can't speak about all these things, so you're only going to get one slide on all new products, and this is by far not a comprehensive listing, but really just designed to inspire some thought and to encourage each of you to make sure that you collaborate with our industry partners downstairs. Share feedback, see what's coming, see what's new, and what we have to look forward to. Likewise on disposables, same thing. I think we're past the point of just having a pump that's going to throw blood forward into a fiber bundle. Right? We need to think about devices that have complete washout every couple of seconds so we have less thrombotic complications. Maybe things that can do atmospheric and hypobaric perfusion. Certainly better coatings, better pumps, certainly non-invasive monitoring so that we have less complications with these circuits. A whole host of vascular access. A new cannula line has just come out from Medtronic designed for longer flows with a tubing that's less thrombogenic. Things like axillary grafts. We heard Dr. Takeda talk earlier about the need to be able to put other things in or tee off an axillary graft. So now having an axillary graft with a side port actually allows us to put a vent in through a side port on an axillary graft. So these are innovative devices that can really help us as we move forward, and we're excited about what's to come. You know, as we think about that, I think we'd all probably agree, though, keeping the circuits simple has really been something as we do more and more ECMO that we've all embarked upon. Having a simpler circuit at the bedside is much easier for bedside caregivers to be able to see what's going on and troubleshoot that circuitry. However, we keep in mind, simpler circuit doesn't mean flying blind. We still need information, and we still need to use that information to take care of folks. So when I think about, and Dr. Agastrand asked me to talk about some of the non-invasive monitoring that we currently do at the bedside and how we use that to help take care of folks, even in a relatively simple circuit. And the things that I think about at first when I think about non-invasive monitoring, that includes things like multiple flow probes with bubble detection, Certainly multiple pressures. We measure pressure non-invasively on all the limbs of the circuit. So that's on the drainage limb before it hits the centrifugal pump, between the centrifugal pump and the oxygenator, and then distal to the oxygenator on the reinfusion limb. And there's a whole host of ways that we troubleshoot, and I'm only going to share one or two examples as we move forward. Things like hemoglobin, hematocrit, arterial and venous saturation monitoring, temperature monitoring, near-infrared spectroscopy, certainly continuous monitoring of PO2 and PCO2 in the ECMO circuit. If we measure the gas on the exhaust of the circuit, that equilibrates almost instantly, so you get a very good and very accurate PO2 and PCO2 of what's being reinfused to the patient. We can do continuous recirculation monitoring, we can do continuous O2 transfer, and we have a whole host of AI-guided troubleshooting that we use to feed information to our clinicians at the bedside to help them do a better job. Now for us, I think using multiple flow probes is important, being able to see flows in every limb of the circuit. Bubble detection is just an added benefit that comes along. So it's not unusual if we have a patient in this particular configuration of VAV with a distal perfusion catheter, we actually have 4 flow probes. So we can monitor flow in every limb of the circuit, including the distal perfusion catheter, and we can predict before events happen when they're moving in a direction we're uncomfortable with. So if we start to lose flow in our distal perfusion catheter, we can usually predict and react to that before we let it thrombose off, and then we're basically causing the need for another operation. We can actually go in and extract clot or thrombus before these things fully thrombose or very shortly afterwards. So there's a whole host of ways that we use this to help us do a better job taking care of folks. Now, pressure monitoring is one of the things that I think is really important, and of course, as I mentioned earlier, we measure each of the limbs of the circuit, the drainage limb, the reinfusion, as well as between the pump and the oxygenator. So this is very valuable when we're troubleshooting, trying to create a differential diagnosis, right? Is this a problem with volume or the drainage cannula, or is there an obstruction? Is there a pump clot or an oxygenator thrombosis? Or is there a reinfusion cannula that's kinked or clot, or is there misplacement? So if we take our gauge bar, and this is our common gauge bar that you see up here on the top right, as we're trying to troubleshoot, we look and we go, well, the venous pressure is minus 31, so at 2.9 liters of flow, minus 31 is pretty reasonable, so we don't think this is a venous cannula issue. If we look at the reinfusion pressure between the pump head and the oxygenator, 260 millimeters of mercury for our particular circuit at a flow of a little under 3 liters, that's a high number, much higher than we would anticipate. So now we know it's definitely some issue that's distal to the pump head. Well, if we look at the reinfusion pressure going back into the arterial cannula, we see that's only 189. So that's actually a reasonable pressure at 3 liters of flow with the current circuit that we use. So we look a little further into the delta pressure, and the pressure drop for 3 liters of flow across this oxygenator, that 71 is really high, and it lets us know we're about to do an oxygenator exchange. So these are the ways that we use non-invasive things like pressure monitoring to help us determine when we're going to need to act before we get into trouble. Now, what about things like hemoglobin, hematocrit, saturation monitoring, temperature monitoring? Again, valuable tools. These tools help us troubleshoot things like patient status, bleeding, oxygenation, oxygen extraction, fever, cooling therapies, drainage, reinfusion. There's a whole host of issues that can happen with ECMO circuits, and we can use non-invasive monitoring to help us. So in this particular case, the hematocrit's 23. It's not great, but not terrible. However, we look at the venous saturation, it's only 51. This is a very low venous saturation. And then we look at the arterial sat. Well, it's 99, so it seems like we're delivering a fair amount of oxygen, but what about extraction? Whoa, the blood temp. So this patient's temperature is 39 degrees. So we have a decent crit, we have a very low venous sat, we have a blood temp of 39 degrees. Luckily, we have continuous monitoring of O2 consumption. This is an O2 consumption index. So for those of you that don't use that, 79 is high, and the 60s is what we would expect. So this patient is extracting quite a bit of oxygen, even though we have adequate oxygen delivery. So anybody have an idea of what's going on here? Right, so this patient is likely septic. So we can then take this information and allow us to take care of patients in a fashion that's appropriate. More importantly, we can set alarms and alerts around this where the computer can actually send a text to my phone that says, hey, Jimmy, Mrs. Smith is exhibiting a septic physiology. You should probably go to the bedside. So we have issues, or we have the ability to actually get notifications, probably even before the bedside care team knows what's going on. Well, sometimes. Maybe they catch it before us. Now, this is one that's probably the newest in non-invasive monitoring for us. This is the ventilation light, which was designed basically for ICUs and ECMOs. This is an electronic blender that was really designed with 5 solutions in mind. This is something that we've been pushing for a long time. It allows us to do patient blending from 0.025 to 20 liters a minute. It has a whole bunch of support and auto-regulation features built in. It has several safety technologies. It integrates several measurement technologies like flows and pressures and temperature. And it also allows us to run a whole host of diagnostics. Now, because of time, I can't spend a lot of time on this, but I'll tell you things like the intelligent gas blending is really fun for us. We can auto-regulate PO2 and PCO2, where a computer can actually adjust our sweep and our FiO2 to maintain those things. We can also take things like hypercapnic patients and say if the PCO2 is over 100, we don't want to bring it down to 35 in the first 15 minutes. We can set over 3 days in equal increments where the computer will actually bring that CO2 down in those increments over a period of time. Whole host of safety technologies that just allow us to measure gas pressures in and out, as well as delivery pressures. So kinked lines, things like that, where in the past we were scrambling, trying to figure out what's going on. We actually get alerts when those pressures go up now. We know exactly what's going on. I'm not going to spend too much time on the diagnostics and the physiologic indices, but just wanted to share with you, we take a lot of these calculated indices, and it allows us to do things like goal-directed perfusion and really come up with strategies to help take care of folks during the course of the run. So I'll just wrap up here with, basically what do our bedside caregivers see? This is one of the screens that we use at the bedside, and this can be as full or as little on the screen as you want. Across the top in those gauges, we put all of our critical parameters that we like our bedside caregivers to see, everything from flows, pressures, saturations, near monitoring, etc. You also see in the center of the screen there the little, what used to be called cannula man, now cannula locator to be politically correct, along with flows and RPMs. And then down the bottom, you see a host of colors there. If you were at the last talk, I spoke a little bit about critical care scoring, very similar to Apache scores that we use in emergency and trauma medicine. Basically, we take several physiologic parameters, we tie those together, and as those things trend in a direction that we're uncomfortable with, we can go from green to yellow to red to let us know things are moving in a direction that we're uncomfortable. We can then click on that, brings up all of those parameters, and tells us what's moving in a wrong direction, so we can really pinpoint our therapies to rectify that. We can continuously see PO2 and PCO2 that's being read from the oxygenator exhaust. We can have continuous recirculation monitoring, continuous O2 transfer displayed, and a whole host of AI-guided troubleshooting tools that we use daily, way too extensive to go into right here. The nice thing is, too, we can see all this information anywhere in the world. So as long as we have an internet connection, we use a network with the hospital, secure network, so we can log on and we can see all this patient information right down to waveforms, parameters, everything, and help our teams even remotely to troubleshoot. So in conclusion, I think the use of automated data acquisition with real-time alerts and notifications, dashboards, clinical decision and reporting tools, non-invasive diagnostics, and a robust informatics system may improve patient care in a number of meaningful ways. And as clinicians, we should consider embracing that non-invasive monitoring coupled with IT and explore the many ways that it can assist us in improving patient care. After all, I believe our patients deserve it. So thank you for your time and your attention this afternoon, and I think we're going to leave questions until the end. Thank you again.
Yes, very interesting, actually. Now it's actually Bob Bartlett who is going to report from his lab. If you haven't never been there, I think you should try to visit it because it's another world and it's so fascinating. You will be surprised what they are doing, actually.
Good afternoon. Kara, the program chair, asked me to talk about innovations in research and allowed 8 minutes to do it. So I'm cutting that down to just preclinical research, which is bioengineering devices and animal testing, and what we do in the lab's a lot of animal testing. So I'm going to present you quite a different type of presentation just to review the topics that we're doing and we think are important. So these are the topics that I'm going to discuss, each one of which we could spend a day on. So eliminating systemic anticoagulation is, of course, a major goal of many of us. Our approach to it has been to create plastic that secretes nitric oxide, which prevents platelets from sticking to the surface. We're into this now, running 7 days with no anticoagulation in animal models. It really does work, and it's it's one approach to surface anticoagulation. But all the other systems, and there are about 8 of them on the market, they really don't work. As you know, you still need some anticoagulation most of the time. You also need to know that there are 2 major clinical studies in the literature, one from Northwestern, one from University of Rochester, in big series of VA and VV patients where they intentionally gave no systemic anticoagulation from day 1, and the results for clotting, bleeding, and everything else are much better than the patients that they anticoagulated in the standard way. So you have to find those in the literature. We have to study this through ELSO. I'm not going to show you any data, but if you just put my name or ELSO into the PubMed, you can find all the papers about this. The second topic is wearable ECMO. Especially with COVID, this turned out to be really important. As you all know, we have patients who are on VV ECMO in the ICU for 2 weeks. Okay, we're used to that. 2 months, 4 months of time. I'm sure you've all had some of these patients. And you'd have one of those, and now you have another, now you have another. So you're using up 3 or 4 ICU beds for months at a time, and patients who are essentially very stable and they don't need to be in the ICU. There are reasons they're staying there now, but there are 2 companies, LivaNova and now Abiomed with the Breathe system, that make devices specifically to get patients up and out of the ICU, eventually home. That's coming. Really important research. In the lab, we're studying the same concept using a system without a pump. This is not new. Other people have done this, but we've refined this to the point where we're now pretty good at it, aimed mostly at babies, actually. Ron Herschel's the PI of this study. He's here. Again, just put his name in PubMed, and you can find out all the data on this particular topic. But it's an attractive way to go for the permanent wearable lung. The device itself is going to have to be outside the patient because you're going to have to change it, but it doesn't require a pump, and it relies on patients with pulmonary hypertension, which of course is a major problem in pediatrics. Also in pediatrics, most of you nowadays are strictly on the adult side, but there's some classic people who've been doing pediatrics for a long time and still are doing it very well. And it works exceptionally well for children, but particularly for neonates. I hope you had a chance to meet Esperanza yesterday, the first neonatal case. But there's a group of patients that's a big problem in obstetrics and neonatology, which are called LGANs, extra low gestational age newborn symptoms, something like that. Basically, babies that are delivered less than 1 kilogram of weight. There are a lot of them every year, depending on whether it's 25, 26, 27 weeks. A lot of them die. A lot of them wind up with brain damage. If you get up to about 30 weeks, almost all those babies do well. They can go on ECMO. They're well managed, but ECMO is not designed for those babies who are living on their open ductus. Their lungs don't work at all. So we've been studying this in the lab for quite a while. George Mikaliska is the PI, making great headway there, and are now at the point in fetal lambs of supporting growth with a version of VV ECMO. We call it the artificial placenta, which works remarkably well. We think that's going to be a major addition to your neonatal ICU in the not too distant future. There's another lab that's working on this, friends of ours at Children's Hospital of Philadelphia. They have a different approach. Our idea is that baby's going to be delivered. Maybe he'll be fine at 25 weeks. A few of them are, but maybe he's going to fail. So we would go to this approach after they fail on attempted ventilation. The CHOP group works via the arteriovenous access through the umbilical vessels, which means that you have to hook it up straight out of the uterus, which means you have to put a woman in premature labor and do a C-section while she's in that labor on the 26th week. And then they're putting the baby underwater. I don't think that's going to amount to anything, sorry. It's just the wrong approach. It's a good idea, great research, but practically is going to have trouble finding its way to clinical. ECPR, of course, we talked about. We've spent a lot of time on this in the laboratory. Alvaro Rojas Pina, who's here somewhere, put his name in. You can read all these papers, done association with Robert Newman, who's the head of emergency medicine at Michigan. And just on the lab side, we've done a series of studies to determine how long you have to wait, what's the best CPR, how about anticoagulation, how about temperature, all that sort of stuff. And while we were doing this, Demetrius Yiannopoulos from Minneapolis, Minnesota, has in fact solved a lot of this problem clinically. We started it at Michigan clinically. We hadn't got it working very well. What Yiannopoulos reports in a randomized trial is about 35% survival compared to basically none for out-of-hospital ECPR. And what he has told us all is that if the heart is dead, don't bother, you're wasting your time. If the heart still has electrical activity, and you all know these patients, they've got resuscitated, oh, we got them back, now they're arrested again. Those are the patients who would benefit from this technology. And finally, we're, for several years now, interested in ex vivo organ perfusion. As you undoubtedly know, if you're involved with transplantation of any organ, you have 6 hours of time from when you take that organ out of the donor patient and implant it into the recipient. So geography is an issue, time is a major issue. You can't go beyond 6 hours. There are cold storage techniques. There are warm perfusion techniques, the Transmedics device you might have seen, but it's still limited to 6 hours. We've been interested in why is that? Why can't you can keep a baby on ECMO for a month with an ECMO machine. Why can't you keep a kidney on an ECMO machine for a month? So we've been studying this quite a while. Again, you can find all this in the literature. But through a very complicated experiment, we got to the point where we can do this routinely for 3 days, and think we could, if you do it for 3 days, you could do it for 3 months. This is the beginning of the concept of organ banks. There ought to be a way to keep organs for transplantation around for at least days at a time to do better matching, to treat diseased organs, to make transplantation elective, and so on. So we're working on that. There are a series of steps that have made it feasible. Hemofiltration is one of them. And we're now developing an automated system that does this automatically. And our next step is to do this with human hearts that are not going to be acceptable for transplantation. We're going to apply the technology to human hearts and hopefully eventually to transplantation. This is not our idea. Our idea is working on it, but this is all comes from a guy named Shaf Khashabji, lung transplant surgeon in Toronto, who has developed this with lungs, can take lungs out, treat unacceptable lungs until they work, and then put them in the patients very successful. So if you want to find that in the literature, look up Khashabji. And that, by the way, the waiting list for lung transplant, he's from Toronto, in Canada is is now a couple of weeks. They don't have a waiting list just because of that. So that's the very beginning of 10 years from now, we'll be talking about organ banks. It's not really like blood banks, but it's going to be a different way of processing transplant organs that you take out of the donor, and then you manage them for a day or two or three, match them around the country. And that's what I see as the future of transplantation of hearts, lungs, livers, and kidneys. So that's just a list of things that we're doing. I'm sorry I didn't show you all the data and the slides. It's easy for you to find if you want to find it. And I think it was 8 minutes. Thank you.
Oh, thank you, Bob. But it is fascinating lab. It will change your mind. Actually, now we have Janine Furch, I think, a neonatologist from Stanford in California. So welcome.
Thank you. What an honor to be here and certainly to follow Dr. Bartlett. I will say, though, if you want a debate of who's going to win the artificial placenta, I'll take up that debate with you anytime. So I'm Janine Furch. I am a neonatologist at Stanford, but I'm also a physician entrepreneur and innovator. One of my greatest joys is getting to teach other faculty members and fellows an innovation process at Stanford Bayer Center for Biodesign. And I've also started my own companies and exited a few. How am I advancing this one? Okay, we figured it out. So as is inherent with all medical device work, I have significant amount of disclosures to make. The most important one is one device that I'm going to talk about today from a company called Novonate. I am their medical director, and I have a small amount of shares associated with that. The reason I'm talking about this device is because it's very simple, but I want to talk to you about how long and how extensive it is to develop even the most simple device. So, you have a great idea. We all have great ideas. But if you're anything like me, it might occur to you, how do I actually get that idea to get to patients? And that's what actually started me on my innovation journey, because I really, really wanted to know the answer to this question. And just publishing wasn't satisfying enough for me. So, as you can imagine, and we've been talking about a lot today, we use medical devices every day, but we rarely think about how they get there. So for each one of these devices, it should be very familiar with all of you. No matter how simple or complex, someone had to have more than an idea. They had to understand the market opportunity, develop a business plan, they had to engineer the device with a team, build a quality system, probably get FDA approval, and sometimes even gather clinical evidence. And each device required a significant amount of time, energy, and capital to be able to reach patients. And it requires a very thoughtful process. And this process we don't often talk about in academia. And sometimes it's even forbidden to talk about in academia, but we need to understand this innovation and commercialization pathway if we want to provide innovative therapies to our patients. Okay, so you're convinced, but where do you start? So you could build your own company. You could share an idea or a product improvement with an established company. You could develop or license intellectual property. You could join scientific advisory boards, or be a consultant or board of directors, or develop local process improvements as well, among just a few things. And so I want to touch on some of those. But first I want to talk about 90% of innovation, and certainly university innovation projects, take emerging technology and then they try to go search around and try to find a need for it. And at Biodesign, we do the complete opposite. We start with a very well-defined need and then use that process and really understand the need to develop the appropriate technology to solve that problem. And so everyone underestimates how long it takes to actually understand a problem, and I certainly did too. But I found that being immersed in this process actually helped my research career as well, because you really, really understand and develop insights to this compelling unmet clinical need. So you do your diligence up front, and then you decide the project that's really worth spending the next 10 plus years of your life. So this is the process with 3 phases. There's identify, invent, and implement phases, and I'm going to go very quickly through this. But the goal is to intrigue you with a brief overview of this proven and repeatable process to structure your innovations. So this picture should be pretty familiar to neonatologists out there and all of you. So adults and pediatric patients have central line management. It's a device, it's protected, it's standardized, it's simple. But this is what we do for neonates. This is actually standard of care. We make a tape bridge out of non-standardized tape, and we put it on an exposed line. And these are our central umbilical venous catheters and arterial catheters. It's non-standardized, it's ineffective, and it's right next to the diaper. So as you can imagine, there's far more evidence of CLABSIs in these lines than in other lines. So umbilical lines, they're the first and most reliable way to gain central access in a critically ill newborn. They're pretty easy to place, they're painless, and then they are often removed in 5 to 14 days, mostly because the fear of malposition or future complications as infections. And so that's background. But what I want to bring you back to is the way that we start to characterize needs, and you can start to think about this, and this allows you to, a prototype to start to talk about what problems you're concerned about, is to put it in a need statement. And this is deceptively simple. So a way to characterize a problem in a population in order to achieve a certain outcome. I'm going to give you a couple tips along the way, which you can take with you. But you'll want to start by defining the problem and then moving it around a little bit, making it bigger or smaller based on the mechanism of action. So here I've listed securing all catheters, securing umbilical catheters. Then you can also consider issues upstream or downstream from your initial observation. So do you want to do prevention? Do you want to do treatment? Do you want to monitor line position? And then this is the hardest part for everyone. So we don't talk about solutions for a long time in this process. So you don't want to embed solutions within your needs statement. And the reason for that is because it really narrows your idea and thinking about what type of solutions could be viable. So for example, a way to prevent CLABSIs is very different, and you can think of lots of different solutions, maybe process improvements and digital health solutions or actual devices versus a way to remove bacterial colonization from umbilical catheters, which focuses you in a little too much. And then when you think about population, you want the more heterogeneous your population is, the bigger the market opportunity, but also the smaller, the harder it is to actually solve the problem with a single solution. So you want to find that sweet spot where your population is homogeneous enough to solve it, but also the opportunity is great enough to spend your time on it. When you think about outcomes, you want to talk about those that are most compelling to key stakeholders and can be objectively measured. I always think about the outcome as the primary outcome of my clinical trial. So do you want to focus on something that's mortality or something that's less valuable like clinician satisfaction? And this group came up with this need statement, a way to protect umbilical catheters in neonates in order to decrease CLABSIs. And then we narrow down and we start to think about all the needs research you need to do in this area. So you need to figure out the disease state fundamentals, a really clear stakeholder analysis. Who is everyone that's involved? It's not just the clinicians, it's not just nurses or people at the bedside, but families in my case and the entire system, existing solutions, and a market analysis. And then we develop need criteria. So we have must-haves and nice-to-haves. Must-haves are essential to addressing the need, and nice-to-haves make the solution more attractive or differentiated. And common categories that we use include efficacy, cost, safety, and usability. And so this is what they came up with. It's called Life Bubble. I'm not showing you this to talk about the device a lot, but it is a very simple silicone device. It's about as simple as it can get, and it's an FDA Class 1 device, so it doesn't even need clinical evidence around it to actually be used on patients. But that device still took a really long time. And they had to think about a lot of different factors, including things like intellectual property and R&D, regulatory strategies, quality management, et cetera. And while they were really capital efficient and stayed in the university for a long time, they also had to go out and raise venture funding to get to 1,000 patients. So it took 8 years for them to do this, so quite a long time. So the point is that if you're going to spend 8 years doing this, you want to do the diligence and effort up front to get the need exactly right. So now that I've convinced you all that you never want to start a company, hopefully that's not true, because it's a wonderful experience too, you can do a lot of other things. In particular, you can share an idea downstairs or a product improvement with an established company. I'm sure they love to hear that. We, in particular, were having difficulty with the negative pressure side of our ECMO circuits and in training air, and so we called up the company that makes that. We knew that we weren't going to start a company around this, and called them up and said, hey, we really want to partner with you to help come up with a better solution. You could develop your own intellectual property and license it out. You can join the board, or you can think of many other potential different solutions. But the idea is that investing in this repeatable process will enable you to use resources efficiently and determine what project is most likely to be successful before you spend the next 10 plus years working on it. So if I've intrigued any of you, there's a free guide at the Biodesign Guide at stanford.edu, and I would encourage you all to go on there, and there's very in-depth multimedia presentations and things along those lines to help you take your idea from an idea all the way to a patient. Thank you.
So thank you for that. And now it is Melanie Bembea from Baltimore Hopkins that's going to talk about It Takes a Village.
Hi, everyone. Sorry, I'm going to move this so that I can see the screen. Cara and the organizing committee asked me to talk about the process of getting an ECMO study funded. And that comes from, for whatever reason, this myth that exists in the ECMO community that ECMO research doesn't get funded, and that it's specifically that it doesn't get funded by the federal government, which hopefully I can burst that myth. And a lot of the work that was just presented by Dr. Bartlett had initially and ongoing funding, significant funding from the NIH, for example. So just to take that out of the way. All my disclosures are research grants, and I have no commercial disclosures. So the challenges that are always brought up when it comes to conducting research in the ECMO population includes heterogeneity of ages and diagnosis, quick advances in technology, advances in clinical practice, feasibility of enrolling critically ill patients on ECMO in clinical research studies, variability in ECMO practices leading to different circuit configurations, different staffing models, et cetera, at different institutions. And again, this question about funding opportunities that are perceived to not be there. So here I'm going to focus primarily on the National Institutes of Health here in the US. The NIH is funding multiple ECMO research topics, studies on topics as varied as circuitry, brain injury on ECMO, care and physiology on ECMO of critically ill children, heart failure-related studies, studies related to hemostasis and coagulation, and multiple studies related to gas exchange, lung transplantation, that sort of thing. So that idea that ECMO research was not being funded by the NIH did come from some pretty sobering numbers. Here you can see the number of projects that are ECMO related that had been funded by the NIH, and you can see there that over a 20-year period there were less than 10 projects. That was followed by the explosion and the really rapid growth of ECMO utilization, especially in adults, between 2010 and 2020, where the number of projects has increased. And just in the last 2 years, there's more projects funded by the NIH that are ECMO-related than you have seen over the last 10 years. So that's really important to keep in mind for anyone who has had any hesitation to submit their work to be reviewed at the NIH. In terms of amounts, dollars, these are all data publicly available, and there's a QR code there for anyone who's interested in looking into this more deeply, in more detail. The peak was there in 2021, and it amounted to all of $8-point-something million. To put this into perspective, there are more than $4 billion going into stroke research, of course, a much larger problem. Over $60 million every year going into cardiac arrest research. So there's room for growth there, and what is needed is to make the case for the significance of ECMO-related research and all the innovation that can occur in the field. The number of organizations is relatively small, with a handful of academic institutions. Following the prior talk, you can see there are a number of companies that are being funded through the Small Business Innovation Research Mechanism and through the Small Business Transfer Technology Research Mechanism, where the latter is typically a collaboration between a small company and an academic institution. And many of these companies that have been funded by the NIH led to production of devices that we're all using in the ICU currently. I was told not to give examples, so I won't, but these are extremely commonly used catheters and other parts of the ECMO circuitry that had their initial steps as part of SBIR and STTR applications to the NIH. And then once that seed funding led to phase 1 development of the device, either they were bought by a larger company or they went on from there to further develop the product through a phase 2, et cetera. So some of the strategies for success are bursting out of our bubbles into multidisciplinary collaboration and not being afraid of getting uncomfortable by working with folks who have expertise that we do not. Engaging in multi-center, international, and multi-network collaborations. Engaging with stakeholders in ECMO-related clinical care, QI, and research. And being more open to novel design for clinical studies, from conventional RCTs to moving to registry-based studies, adaptive or stepped wedge design. We've heard a little bit about that yesterday. Other strategies for success include existing registries and infrastructure. There are lots of large national and international registries that have been primarily developed for QI purposes, but that include large patient populations at risk for critical illness requiring ECMO support. Also, as we heard yesterday, it's really important to have a denominator. So, of course, all of these can be then applied for ECMO research. And joining research networks. And some of these are only adult research networks. Many of them are pediatric research networks. In some ways, in pediatrics, we're forced to collaborate a little more because we do not care for a very large number of patients at a single institution, and a single center study is bound to be very small and underpowered. Setting an ECMO research agenda as part of this large research consortia, pursuing funding and not being derailed by this sense of pessimism. And that applies not only to NIH funding, but it also applies to other federal funding here in the US, for example, at the Department of Defense. the AHRQ, and also with various societies and foundations. And establishing ground rules for data guardianship, data coordinating centers, and statistical support. That's all I have, keeping to the 8 minutes. And I think we're going to take questions next. Later, okay. Thank you.
Thank you so much. And now it's Nicole Gillette from discussing about FDA perspectives. Thank you.
Hi, I'm Nicole Gillette from the FDA. I'm an assistant director in the Circulatory Support Devices team, and I'm going to be talking about our perspective on innovation. I have no disclosures. So first, I think it's really important that we level set. And so there are multiple ways to US patients through the FDA. First of all, to do a clinical study, if you need to get prospective data that's not already out there, you can do an investigational device exemption for your novel device. These are clinical trials for significant risk devices, but they can also be studies for research, publications, or to support marketing approval or labeling claims. And they can also be prospective registry embedded trials. And then once you have your clinical data, or if you're going to use some real-world evidence, then you would go into your marketing applications. It's important to note that a 510K is a Class 2 device, and 510Ks don't always require clinical data. So it's important to come talk to FDA to figure out if you need clinical data or not. For ECMO, I can go ahead and tell you that you're going to need some clinical data. So 510K, it evaluates substantial equivalence. It's looking at a clearance. De novo, that means that you have something novel, either a new indication, a new device, but we think that it can be regulated in the Class 2, you just don't have a predicate. So we're looking at safety and effectiveness there, and then once that de novo is granted, it would become the predicate for future 510Ks. You have PMA and HDEs, which are Class 3 devices, and there we're looking at safety and effectiveness, and you'll be approved if you meet the bar. All right, so when we're talking about devices in the ECMO space, it's really important for FDA that we clarify because we have two different sets of regulations. So for cardiopulmonary bypass, which we defined as anything less than 6 hours, we're looking at oxygenators, CPB cannulas, non-roller blood pumps that are used for less than 6 hours, and these all have something similar of a limitation of 6 hours in their indications for use statement. You look at ECMO, you're going to see a lot of similarities: oxygenators, cannulas, systems, blood pumps. However, the difference is that at FDA, ECMO is greater than 6 hours. And so for those, you're going to see the limitation. You won't see a limitation if they've been tested for more than 7 days. So it's important to note that while the device may be the exact same device, the indication would drive the regulation here. And that's going to be important when it comes to how you want to market your device and what testing you're going to need. And then we have to get a little more complicated because ECMO, it was reclassified in 2016. And so what this means is it used to be Class 3 and you would need a PMA. And then in 2013 and 2014, FDA convened 2 panels that evaluated the data that was out there. And ultimately in 2016, we came out with what was the outcome of that final order. And we decided that there was enough information that components or systems indicated for patients with acute respiratory failure or acute cardiopulmonary failure where other available treatment options have failed and continued clinical deterioration is expected or the risk of death is imminent would be a 510K for ECMO. We also went on to list a couple different indications that we thought there was enough data to support a 510K for. A couple of those are meconium aspiration in neonates and pediatrics, failure to wean from CPB following cardiac surgery in pediatric and adults, and ECPR in adults only. So then you kind of wonder, well, what's not in the final order and what do I do with those? And so the final order is explicit sometimes of what was not included. So high-risk PCI, traumatic resuscitation, failed heart and lung transplant, acute ARDS, acute decompensated of COPD are not part of that final order. So since the final order, FDA and you all as the community have been making a lot of strides into getting devices on label that have been used off label. And so since the final order was issued in 2016, there are now 7 clearances for ECMO indications. Only 7. So we have a lot of work to do. The other current regulatory landscape that's important is that in the COVID-19 pandemic, we understood that you needed the access to these devices. So remember, a couple slides ago, there was the CPB devices that can be used off-label for ECMO because FDA doesn't regulate the practice of medicine. So FDA released a guidance document that described our enforcement policy during this public health emergency of COVID, which allowed companies to talk about ECMO even though they were only cleared for CPB, just for COVID though. So the current regulatory landscape, if you talk to anybody, is that we're encouraging these CPB cleared devices that can be used safely for ECMO to work with FDA on getting ECMO on label, because we know devices that are used on label are ultimately going to be safer for the patients. So you got to figure out if it's a 510K or not, and FDA loves our guidance documents. So we have a lot of guidance documents to talk about the 510K program. This flowchart, which I don't expect you to be able to read, can easily be found. But ultimately, what it's walking through is to figure out, is there a predicate legally marketed? And so that predicate device could be a cleared ECMO device, or right now, while we recognize that there are not a lot of cleared ECMO devices out there, you could utilize the final order as your predicate, which is a little novel. If you keep going down the flowchart, and let's say you decide, we do have a predicate device, and you have to answer a couple other questions. One of the tricky questions that people sometimes get confused with is that they think that they can use data to show that the different characteristics do not raise questions. And that's not true. We need to get through this question on if there's new characteristics, new questions of safety and effectiveness, before we can get to evaluating the data or bringing in a reference device to show that there aren't any questions. So help, my indication is novel, I don't know what to do. Like I said, FDA has a lot of guidance documents. One of them would be general to specific. So we'd recommend to consult that and see if the intended use is the same, as well as endpoints and other characteristics in that guidance document. And if all of that aligns, then good news, a 510K may be appropriate. But if not, it might still be a Class 2 via de novo if the risk can be adequately controlled. And one example of somebody going through a de novo process was the Hemo-Lung Respiratory Assist System, which got a de novo granted last year for ECOR, because that was outside the final order, but it now has its own regulations. So what do you do if your design is novel? Well, you might have new technological characteristics, but maybe the differences don't raise new questions. And if so, then you might still be a 510K. And one of these examples is Spectrum Medical's heater cooler, which was cleared earlier this year, which uses a novel fluid. We decided that didn't raise new questions, so it could still be a 510K. So in summary, transitioning CPB devices for on-label for ECMO where appropriate is important for patient safety. New indications and design are encouraged for both CPB and ECMO to make the device safer for patients. And novel does not always mean a PMA. We always encourage you to come talk to FDA early in the device design process to determine the recommended regulatory pathway. Thank you.
So, thank you so much. And now I think we have the panel discussion, so the speaker can come up. And many of these questions is a little bit not familiar for me because I come from Europe. So FDA and NIH is nothing I know so much about. So you have to come up with the questions, please. Someone? It's hard for me to come up with. I can actually start with James Beck. These devices you show, do you have to recalibrate it over and over and over again?
So, that's a great question. Yes, some of the devices do need to be calibrated, the sensors in there. So some of the new technology uses the Massimo sensor technology, and that involves really just changing lines. But there are some sensors that need to be recalibrated to ensure accuracy, especially since we have computers helping us make decisions, and clinicians for that matter, making decisions based on these values.
And you said, I think so you said something really, keep the circuit simple. I think we really have to think about that. But then you said something bad. You could have it on internet connection so you can see it from all over the world. Can you somehow take that person to move him bedside? So he doesn't sit in another room and take decision. He should go bedside. Everything you make that move people from the bed is bad. If you can make them go to the bed, is good news.
I absolutely agree with you, and I'm glad that you said that I said something bad, because my mother would probably have examples that were much more impactful than that. But you're absolutely right. So the fact that we can see it remotely doesn't really, we don't use that to help us move away from the bedside, but we use that to help us see what's going on and get our clinicians to the bedside faster, and then help them remotely. So when there's issues or when they're having trouble understanding something, I can see what's going on and talk to them while they're at the bedside. So we actually use it to get to the bedside faster to take care of folks. And I absolutely agree with you. There's nothing more valuable than being at the bedside to assess what's going on.
Good news. Bob, do we ever have a system without heparin? Ever? Does it? We started with Carmida coating surface for 35 years ago. Nothing has happened since then. And how does it really look in the next 10 years?
Quite right. Well, of course, that's what we're aiming for. There are a few new surface coatings that are not in our lab that I didn't refer to that we're studying there. The nitric oxide idea looks very good. So it works in animals, but of course that doesn't mean it'll work in patients. So that's where we're going next. But I think that in time we will be able to manage ECMO without systemic anticoagulation. And I encourage you all to find those two papers that I referred to, one by Bharat, one by Prasad, where they intentionally gave no intravenous systemic anticoagulation for entire runs in 70, 80, 90 patients with less bleeding and clotting complications. It's really important, and I've been encouraging those guys to come to ELSO and let us organize a study through ELSO where we have centers that do that, compare to their data, and see if it's as good as they say. I must say, I believe it's probably the case. Now, both of those studies involved giving the patient 5,000 units of heparin sub-Q 3 times a day, so-called DVT prophylaxis. So that may or may not be important, but it's nothing like systemic anticoagulation with the problem. So if you're interested in this topic, do search those two papers, send them all an email and say, please study this in an organized fashion.
And another question is, you talk about this really small neonates, 500 to 1,000 grams. It was paper for a long time ago that said that even with small heparin doses for arterial line make more brain bleeding than before. And now we should put them on ECMO.
No question, but that's where the nitric oxide comes in. So in our studies in premature lambs, I emphasize it's lambs, they don't have any lung function, but their brain bleeding is less than humans. But using the nitric oxide system as an example, we can do this without systemic anticoagulation. We've done it for as long as a week, and it seems to work. So that you're quite right. We want to avoid systemic anticoagulation in those very premature infants because the risk of intracranial bleeding is particularly high.
And then you talk about organ perfusion. When I was in your lab in '89, we actually took a kidney on ECMO. Everything was perfect, and the kidney died after 7 hours. And then we had the dog there and the kidney there, and we mixed the flow 1 minute per hour. And the kidney survived 22 hours. Has anything coming on from that?
Yes, a lot has changed over time, but Polly, you're quite right, of course. If you take a kidney, heart, lung, liver, and perfuse it with an ECMO circuit, which ought to make it last forever, it always fails within 12 hours, let's say. So the question is, why is that? So our research has been focused on the scientific topic. why is it that we can't perfuse organs? We have a theory that I think is right, I don't know if anyone else does, that has to do that there's a unnamed brain hormone that maintains capillary integrity. That's our hypothesis. We will see if it works. But we're up to days of time now following that idea.
So you haven't found Ritalin yet?
Yeah, Polly knows we call this vital, and there's a pretty good rationale for it. Again, it's in the literature. You can look it up.
Yeah. Okay, good. Janine, this is very complicated for me, but is it a big risk if you come up with a good idea, you go to another company and ask them, the risk that they just take the idea from you? Must be big.
So this is often...
How do you protect yourself from it?
How do you protect yourself? So this is often, a very large concern of clinicians and individuals. How I look at innovation is a little bit different, and maybe that's because I'm a neonatologist and we don't have a lot of innovations and we absolutely need them, and they're not always incredibly valuable in our field, although some certainly are. It's a team sport, and you have to figure out your team sport. If you're not going to do it, but you really need it for your patients, or you've researched it, you can publish on it, you can do something along those lines, then there are certain times where it makes sense to partner with a company that already has the entire engine and team and everything that you need to make that go forward. I always tell my students, and this is a bit controversial, but ideas are worthless. Execution is everything. And so they get a little bit mad at me when I tell them that, but and it's not entirely true, but your idea is not worth anything unless you execute on it. So let's figure out a way that we can execute on things and bring the best care to our patients possible.
And it takes 8 years, so you have to be very sustainable and going on before otherwise it fade away?
There's, it can take between 5 to 20 plus years to get a device out there. And that's when you've already done the background science to it as well. So it certainly takes a quite a long investment. The reason I gave you the 8 years idea was because it's an extremely simple device, but it still took that time to build the entire structure around it. So it's not an undertaking for the faint at heart.
Okay, thank you. Now we come into the NIH. I can have help me, whatever. Come on with the question, yeah?
This is a question for Nicole Gillette. Can you tell us how much longer the EUA will be in place?
So I think you're talking about the immediately in effect guidance document that's allowing enforcement policy for COVID.
Yes.
Yeah, unfortunately, I don't have a magic date of when that guidance document is going to be withdrawn. Our hope is that there will be ample public notification, and it would be published on our website whenever they start talking about that. The guidance document is in effect as long as the United States is under a public health emergency, which we still are for COVID.
Okay. I mean, is
More
it
to come.
Weeks or months? Is it on that order?
I truly have no idea.
More questions, please? Oh yeah, come up. Oh, you need a mic. I'm deaf.
As a Michigan alum, it's always great to see Dr. Bartlett in person, and it's always an honor to be here. This question is for the gentleman from Columbia. I apologize, I was a little bit late and missed your name at the start.
Jimmy.
Jimmy, thank you. I'm Ewan Wong. I work at Memorial Regional Hospital in Hollywood, Florida. So my question is that you gave us a really very insightful operations to your program with the use of these various monitoring devices and even ultimately incorporation of AI. My question is, what is your current bedside staffing model, and what do you expect the staffing model to transition to as you incorporate more and more of these advanced devices and monitoring process?
So I think that's a great question as well as a great concern. So right now our bedside care model is we have one nurse to one ECMO patient at the bedside. There's no ECMO specialist and there's no perfusionist at the bedside. We have a perfusionist in-house 24/7. All of these things are being monitored remotely from a pump room location as well as from phones. We train our nurses. One of our perfusionists, Dana, that gave a talk yesterday, she does all of our training around ECMO in-house. So that includes, physicians, respiratory therapists, physical therapists, nurses, etc. So they are trained to be able to deal with emergencies like line separations or loss of gas and things like that. They immediately call a number. We still use a phone, 317 ECMO. You can call that from in-house or out-of-house. You get a perfusionist 24/7, and we respond to the bedside. Will that model change? I'm not sure. Right now we have one person covering up to sometimes 20 ECMOs simultaneously. We also duplicate that model, however, on our children's side. So our children are all in one unit, and we have a pump room close to that unit, so we're not sitting at the bedside, but again, one clinician monitoring, several ECMOs working with the bedside nurse.
I think this training is very important, actually. In Stockholm, we actually train our staff 4 hours for every 5 weeks. Every individual person has to do that for emergencies like you talk about. So everyone in our unit will change an oxygenator or pump head within 30 seconds. Otherwise, you cannot work there. 30 seconds. We are prepared for it. We really have everything around us that can do it. And that means that makes a big difference when you are in the room, because suddenly when you look at the machine and you know that everyone can do this is suddenly not an enemy anymore. It's your best friend in the room. As long as the pump goes, nothing will happen with the patient. So if you can come up to that you start to feel inside your heart that is my best friend and not the enemy, you start to think totally different about everything you do with the patient. It's a major step to come up to that. But also, we have an ECMO ICU. We do neonates, pediatric, and adult, and nothing else but ECMO. So it's easier for us, probably. I think that. More questions? NIH funding, I can nothing about.
It's all good, no worries.
So we stop there, I think. Thank you so much for listening. Thank you all.